Hello students, in our last lecture we had discussed about overview of operon and structure of operon, and we have seen that there are two kinds of operon, inducible and repressible, and today we discuss about Lac Operon, example of inducible operon. In 1961, Jacob and Monod they described the operon model to study the genetic control of lactose metabolism in E. coli. Lactose operon, that is designated as Lac Operon, codes for enzymes involved into the catabolism, that is degradation of lactose. Lactose is a disaccharide which is made up of glucose and galactose. It is an inducible operon since in the presence of lactose induce the operon to switch on. So it is a negative inducible operon in which allolactose act as inducer. These operon consisting of three structural genes Lac Z, Lac Y and Lac A. Apart from structural genes, it consisting of repressor gene that is Lac I. It consisting of promoter Lac P and operator region Lac O. Three structural genes Lac Z, Lac Y and Lac A encode for three different enzymes for lactose metabolism: beta-galactosidase, galactoside permease and transacetylase. The repression of Lac Operon completely shut down transcription. This is the structure of Lac Operon. This is a repressor gene Lac I. This is the promoter region and this is the operator region, which is known as a regulatory region. And three structural genes are there: Lac Z, Lac Y and Lac A, in which Lac Z encode for beta-galactosidase, Lac Y encode for the enzyme galactoside permease, and Lac A encode for the enzyme transacetylase. You can see over here Lac Z break down the lactose into glucose and galactose. The enzyme is beta-galactosidase. The enzyme galactoside permease encoded by the gene Lac Y. It is a protein which is found into the cytoplasmic membrane and responsible for the transport of lactose from outside of the cell to inside of the cell. And the third enzyme, galactoside transacetylase, encoded by Lac A gene, the exact function of this gene is not known. Now, when lactose is present, basically in the operon structure also we have discussed that the product of regulatory gene binds with the operator region. In the absence of lactose, means into the normal circumstances, as it is an inducible operon, we had discussed that they are usually shut off. Means operon is off and no transcription takes place. You can see in this picture that in the absence of lactose, the product of regulatory gene is active regulatory protein, that is active repressor. This active repressor bind with the operator region. As operator region and promoter region have overlapping sequences and operator region is occupied by the regulatory protein, RNA polymerase cannot bind to the promoter, and so no transcription of structural gene takes place, which is in turn not required. Why? Because lactose is absent. So in the absence of lactose, the regulatory protein binds with the operator and inhibit the process of transcription. Second condition, in the presence of lactose. When lactose is present inside the cell, some of the lactose is converted into allolactose. These allolactose act as inducer. These inducer bind with active regulatory protein and make it inactive. So, in the presence of lactose, active regulatory protein is converted into inactive regulatory protein. This inactive repressor or inactive regulatory protein cannot bind to operator region. So RNA polymerase will bind to promoter and it will carry out the transcription of structural genes. So the structural genes are transcribed, translated and synthesized three different enzymes for the lactose metabolism, which is in turn required. Why? Because lactose is present. So this is a kind of regulatory mechanism where the regulatory sequences, promoter and operator, it will decide whether the operon will be turned on or whether it will be turned off. This is known as example for negative control of inducible operon. Now the Lac Operon has second layer of also control mechanism, which is known as a catabolic repression. That is also known as a positive control. So we can say that there are two kind of control mechanism in case of Lac Operon. One is negative control and second one is positive control. Negative control we had discussed in two conditions, in the absence of lactose as well as in the presence of lactose. Whereas the regulatory protein decides whether the structural genes will be transcribed or will not transcribe. The second control mechanism is catabolic repression. Catabolic repression is the ability of glucose to prevent the expression of genes. In bacteria, it is a kind of positive control mechanism. In this mechanism, a protein known as catabolic repressor protein, that is CRP, which is an active protein that binds to the target sequence at the promoter. This CRP binding plays a significant role to control the expression of genes into the operon. It's a positive control mechanism. The positive effect is activated by catabolic activator protein also known as cyclic AMP receptor protein as CRP. This CRP increases the affinity of RNA polymerase to bind the promoter at certain sequences that are known as regulatory sequences located near to the promoter sequences. Now CRP is an inactive protein and its activation requires cyclic AMP. When the CRP is in inactive form, it cannot bind to the regulatory sequence, and so that there is no affinity of RNA polymerase to bind to the promoter and no transcription takes place. But when the CRP converted into active form with the formation of active cyclic AMP CAP complex, it binds to the regulator and increase the affinity of RNA polymerase to binds to the promoter. So it plays a significant role for transcription to get turned on. So in this entire mechanism of catabolic repression, CRP plays significant role. And the CRP binding, it entirely depends upon the cyclic AMP, which is CAMP. So let's see what is CAMP, how it is formed and how it activates CRP and what is the role carried out by activated CRP to control this mechanism. Now the CAMP, that is known as cyclic AMP. It is generated from AMP by the activity of enzyme adenylate cyclase. CAMP, known as adenosine 3'5' cyclic monophosphate. The concentration of CAMP is inversely proportional to the concentration of available glucose. Means you can see that when the glucose level is high, there is high amount of ATP. And ATP is converted into AMP. AMP is converted into cyclic AMP by the activity of adenylate cyclase. But high ATP affect the activity of adenylate cyclase. Means high amount of ATP means the low adenylate cyclase activity and there is less cyclic AMP production. Whereas when ATP is less, then the adenylate cyclase activity is high. So high amount of cyclic AMP present. This cyclic AMP bind with CAP and the formation of CAP cyclic AMP complex takes place. and it binds to the site which is located slightly upstream for the lac gene promoter, which is at the regulatory sequence. So concentrate, high glucose means high ATP, means high AMP, less adenylate cyclase activity and less cyclic AMP. Whereas there is less glucose means less ATP, means less AMP, there is high adenylate cyclase activity and high amount of cyclic AMP. Now you can see over here promoter, slightly upstream to the promoter there is a activator binding site. It is another kind of regulatory sequences present into the operon. Inactive activator means inactive CRP, to which when the cyclic AMP bind there is a formation of active activator means active CRP. This active CRP binds at activator binding site means at this particular sequence. The binding of CAP cyclic AMP complex at this particular sequence increase the affinity of RNA polymerase to bind to the promoter. So RNA polymerase quickly bind to the promoter and carried out the transcription of the genes for the metabolism. You can see over here when there is an inactive CRP, no transcription takes place. But when the CRP gets activated, it increases the affinity of RNA polymerase to bind and transcription gets started. So, when you provide cells with lactose and glucose both, cell will utilize glucose first. Means inside the medium when there is a high level of glucose, you can see into this lower mechanism, when there is a high level of glucose, there is a less level of cyclic AMP. We have seen into the previous slide, it is a irreversible proportional mechanism. So in the presence of high level of glucose, there is a level of cyclic AMP is low, and so that there is no CAP-CAMP complex formation takes place. Means we can say that CRP is into inactive form. This inactive complex cannot bind to the regulatory gene. It cannot bind to the active site for the activator protein binding, and so that RNA polymerase cannot bind to DNA very efficiently, and so that there is little or no transcription takes place. But as glucose level is utilized inside the medium, there is a low amount of glucose, and in these circumstances the cell will switch over to the use of lactose. What happens when the glucose level is low? The level of cyclic AMP are high. This cyclic AMP rapidly bind with the CAP, or you can say CRP, and form CAP cyclic AMP complex. It is also known as an active CRP complex. When this active CRP complex binds at the activator binding site, it increases the affinity of RNA polymerase binding, and so that RNA polymerase quickly bind to the promoter region and transcription takes place. It is a two-way mechanism. Apart from that, concentrate, when the glucose level is low, lactose is already present inside the medium. So, lactose serves as an inducer and it prevents the binding of regulatory protein to the operator. So, it's a kind of two-way mechanism. Lactose, not this active repressor, cannot bind to operator. So, promoter and operator, both the regions are free, and this active CRP protein increase the affinity of polymerase to bind to the promoter. So as a result, high level of transcription takes place and it is followed by translation, and all the three structural genes are transcribed, translated, and enzymes are synthesized for the lactose metabolism which are required. So this is known as catabolic repression. That is, in the presence of glucose as well as lactose, the cell will utilize glucose first. And when the glucose level decrease to a certain level, the cell switch over from the use of glucose to lactose. So this is all about Lac Operon, which is example of inducible operon. We had discussed two kind of mechanisms in Lac Operon, that is negative inducible control mechanism, and second one is a catabolic repression. The next operon will be the tryptophan operon, that is an example of repressible operon that we will discuss in our next lecture. Thank you very much.